Welcome back, ladies and gentlemen. Today we are going to wrap up our discussion of Chapter 7 and membrane function by talking about active transport. So up to this point, we've talked passive, so moving from high concentration to low concentration and not requiring energy to do it. Well, active transport is, for the most part, the exact opposite. So now I'm going to require energy in order to move materials against their concentration gradient. So passive always went with it or along the concentration gradient, high to low. Active transport now is going to require the energy to go from low to high. And if you think about it, hopefully that makes sense, right? So passive movement is the path of least resistance. So everything kind of just spreads out, oozes out. Um, if you leave your bedroom go long enough, right, it just kind of turns into chaos because you're not expending any energy in order to maintain order. Active transport is that maintenance of order. So there are situations where your cells absolutely need to maintain those concentration gradients in order to perform some type of cellular work. But in order to keep that order, that's going to require energy. So to clean up the mess, I need to expend energy to do it. And your cells are typically going to use ATP as their energy molecule to run active transport. The other thing with active transport is that it will absolutely require a specific protein embedded in the membrane in order to move things, regardless of their chemical properties. Now, typically speaking, active transport is only really going to be effective on those hydrophilic materials, right? Because anything that's hydrophobic can readily pass across that membrane, and I'm not going to be able to maintain that concentration as a result. So active transport's only going to be able to maintain concentration gradients for anything that is hydrophilic. So any sort of ions, any sort of polar molecules, those are the things that I can continually build a concentration gradient and maintain a concentration gradient using active transport, because this is material that can't just readily pass across those phospholipids. So one of the most important and most common active transport systems in animal cells is your sodium-potassium pump. We're going to come back to talking about these when we get to your nervous system because they are incredibly vital to allowing your membranes to have a potential on either side of those nerve cells. So we need that electrical potential in order to send signals. If we just allowed diffusion of ions, for example, to occur, and everything equalized out, you wouldn't have the ability to send any nerve signals, and that's not good. So how does your sodium potassium pump work? Well, it is naturally configured to be open to the inside of your cells, and that's what we're looking at right here. And they are going to load 3 sodium ions from the internal environment of your cell every time that this works. So once those sodium ions, I should say, attach to that protein, we add a little jolt from ATP. Essentially, we do a hydrolyzing chemical reaction. So water comes in, we pop off that phosphate, and that excess energy that gets released as a result of that removal of that phosphate from ATP, we can use then to plug that in and phosphorylate that protein. Once that phosphate is attached, that forces a shape change to the protein. What that means is that that shape now opens the protein to the outside of the cell. In that process of now opening to the outside of the cell, it also changes the bonding sites for those sodium ions, and they can no longer stay bound to the protein. So they get released out into the cytoplasm. So when we look, we've got relatively low concentrations of sodium on the outside, high concentrations on the outside of my cell. So low inside, high outside. That's why this is active transport, because I'm moving it from where it's already low to where it's already high. Now, so long as that phosphate remains attached, that protein stays open to the outside of your cell. That's going to allow potassium ions to attach. So sodium gets released, and now we're allowing the shape change to those potassium sites to be readily accessible to those potassium ions. So those potassium ions load. When they do that, they cause a shape change as well that kicks off that phosphate group. That phosphate is now free to go back into the cytoplasm and eventually get reattached to your ADP to make more ATP. And we'll talk about cellular respiration later this year and how that occurs. But for now, it's just off it goes, right? So once we lose that phosphate group, that causes the protein again to open back to the inside of the cell, also changes the bonding sites for those potassium ions and forces them to be kicked out of the protein. They're now released to the inside of the cell. We start the cycle over again. So if we look at those potassiums, right, I've got low potassium outside my cells, high potassium inside. And so that's where the active transport system comes. And then the whole cycle keeps going. And so we use that as a way to maintain certain gradients of electrical potential in your nerve cells, which is really important. We can just have general proton pumps. So speaking of that membrane potential, it can be a pump like that sodium potassium pump, or there are other proton pumps that exist. But essentially what we're saying is we're creating a voltage difference across the membrane so that essentially I get a more positive charge on one side, more negative charge on the other. Some of them can be proton pumps, specifically talking hydrogen ions. We can use other ions like the sodium potassium pump in order to create those membrane potentials, but we're creating an electrochemical gradient. And that is done by causing the concentration of those ions to differ across the membrane. And in order to do that, I need to be able to force things in one direction or the other, hence the necessity for the ATP and the energy. When we get to cellular respiration, we'll talk about some proton pumps specifically that are embedded in your mitochondrial inner membrane that are going to allow us to set up a hydrogen ion gradient, kind of like what you're seeing here, that's going to be able to drive the ability for us to make lots and lots of ATP, which is pretty cool. So stay tuned. These guys will come back for there. A lot of those proton pumps, speaking of cellular respiration, are actually part of co-transport systems. So those proton pumps that I mentioned with the cellular respiration, if you've heard of your electron transport chain, that electron transport chain is a bunch of proton pumps. And they actively move hydrogen ions from the inside of your mitochondrial matrix, or the jelly-like substance in the middle, and push them into that inner membrane space. So there's space between the two membranes of your mitochondria. And we set up a hydrogen ion gradient. That works as a co-transport system to allow us to let that hydrogen diffuse back through an enzyme called ATP synthase. And we use that then to attach the phosphate groups that are floating around to ADP in order to recreate ATP. Now here, this is showing you a similar idea, but rather than it being the cellular respiration process, this is in your cell membrane where we're using those proton pumps, pumping those hydrogens out into that extracellular space, and then using the diffusion of that gradient that we've created to do work, in this case, to bring in sucrose, to bring in sugar. So it needs a little extra oomph in order to get that sucrose through that membrane. We don't want to expend the ATP that it would take to get the sucrose through just by itself. We can make a more efficient system. We've evolved, I should say, a more efficient system by using minimal ATP to pump the hydrogen ions out here. That gradient then gives us a little extra oomph in order to get that sucrose through. So we expend less energy doing it in this respect as opposed to what it might take to bring that sucrose in all by itself. And these are pretty commonly active systems to drive those nutrients into cells as a way to more efficiently get it in without expending more energy than is necessary. So pretty cool system that we've managed to evolve over time. Okay, so let's talk bulk transport. This is also a form of active transport in the sense that I'm going to require energy in order to do it, but it does not necessarily follow the path of low concentration to high. So the only reason we throw bulk transport here into active transport is because I need the energy in order to make the vesicles that are going to get produced to bring these large particles in or out for that matter. So we talk exocytosis versus endocytosis, and if you know your prefixes, then you should know which is which. So exocytosis, this is something that the cell has produced, whether it's a waste product or a hormone or something else that needs to be used somewhere else in the body. And those transport vesicles migrate to your cell membrane, they fuse with it, and they spew their contents out of the cell. So a lot of secretory cells, glands, the ends of your synapses in your neurons, releasing neurotransmitters, all of this stuff is occurring via exocytosis. So remember going back to that endomembrane system, right? Nucleus contains the instructions, sends those instructions via the RNA molecules to the ribosomes. Ribosomes produce the proteins. That rough ER then packages them into vesicles, sends them to the Golgi. The Golgi modifies and packages and ships into another vesicle. And if that material is meant to leave the cell, then this is what's going to happen. It's going to go to the cell membrane, merge with it, and release the contents. So where does the energy come in? Well, the energy comes in moving these vesicles around your cells. Endocytosis works in the opposite direction then. So endocytosis is now going to bring in large molecules. Something like when we talked about those big phagocytotic cells in your immune system, right? They're bringing in that bacteria via phagocytosis. Literally, the cell is eating those big bacterial cells and then they'll break them down, right? So phagocytosis is cell eating. If they're more liquid particles, like we were seeing here, then we talk about it as penocytosis. So endocytosis is a general category of bringing any large particle in. Again, it's going to pinch around, make that vesicle, and bring it in. If it's large physical stuff, solid stuff, then we talk about it as phagocytosis. If it's liquid material like we see here, then we're talking penocytosis. Remember that this food vacuole then needs to go merge with a lysosome in order to break this material down. So there is some connection back to that endomembrane system yet again. We also mentioned here and there about this idea of receptor-mediated endocytosis. And so your cells in certain situations have these receptors that sit on your cell membrane, and once they are activated based on ligand molecules or solute particles, whatever the case might be, that will actually trigger the endocytosis. So in some cases it just kind of happens, and in other cases there is very specific lock and key ideas in order to bring this in. And this is often how animal viruses make their way into our cells because they utilize the connections of their antigens to our receptors. And that's how they bring themselves in, they uncoat, and then they take over the cell. And then when they go to actually release themselves rather than just blowing up the cell, they'll actually take some of those pieces along with them as they pinch off some of our cell membrane to make their new envelope, which is pretty crazy. So just wrapping it all up here, right? We've got passive transport, so moving things from high concentration to low. I don't need energy to do it. And depending on the mechanism and the material that I'm moving, it can be simple diffusion, it can be osmosis if we're talking water, and water, technically speaking, is a type of facilitated diffusion. And so that diffusion can be facilitated, helped out by those proteins, either by channel proteins or those carrier proteins. Your active transport then is going to require proteins in order to move those solute particles. But now I'm going from where I am low in concentration to where it's already high. I'm forcing the concentration gradient to continue to exist rather than in passive transport where if we leave it go, eventually it'll reach equilibrium and even out. But in order to maintain that gradient, that means energy has to get expended, used up in order to maintain that process.